try to make this work. So thank you, everyone. My name is Tracy Lively. I work at the National Cancer Institute. And just to be very clear about what my vantage point is here, thank you for inviting me to be part of your meeting. At the NCI, I am part of the clinical research community. I'm a PhD, I'm not a physician, but I work every day to try to support the development of better diagnostic tests. Usually the first thing I have to explain about my job at NCI is, even though I work in a program called the Cancer Diagnosis Program, we're not the cancer imaging program, so our radiology colleagues work down the hall from us. We focus on the development of the kind of diagnostic tests that might be ordered by an oncologist to be performed by a pathologist in a clinical lab on some sort of sample like a blood sample or a tumor specimen. We collaborate very closely with our colleagues in the cancer therapy evaluation program. And so, again, to clarify, the kind of diagnostic tests that we focus on are those that would be most useful for planning treatment for someone with cancer. We have an entire division, the Division of Cancer Prevention, that is fostering research into the kinds of diagnostic tests that would be useful for screening in the early detection setting. And the other thing that we do as we work with our colleagues and with the researchers that we support through research grants is to facilitate sort of a conceptual development in the process of diagnostic test developing. And so that's going to be the focus of my talk for the next few minutes, and I hope it will help set the stage for the talks that we'll hear next in our panel, where we'll be looking at some of the more specific kinds of tests. Okay, so for starters, what is a clinically useful diagnostic biomarker? Patty's slides had one definition in them. This is another one that I've seen used quite a bit. It was devised by a panel convened by the FDA, and it's published, so I guess it's sort of an official definition. Something you can measure, something that indicates something that might be normal or abnormal. But this is a different way of saying it that I lifted from a slide years ago that was presented by a physician, and I think this is actually what we're after. Readily interpretable and useful information. It might be information to guide choosing a course of action in therapy. It might be information about how well a treatment is working. And so what's the framework? I think many of us here are very familiar with the conceptual framework that we use in drug development for oncology. So we use a phase 1 trial to look for a dose. We use a phase 2 trial to look for signals of anti-tumor activity. And then in a phase 3 trial, we're trying to actually establish how well the treatment works and hopefully set the stage for an approval by the FDA. This is a conceptual framework, and even if we don't follow it exactly every time, it's still there to guide our thinking about drug development. When it comes to diagnostics development, I'd have to say that we don't yet have such a solid conceptual framework, but it's emerging. This slide has on it one of the very influential publications that has introduced the concepts of analytic validity, clinical validity, and clinical utility that are important in diagnostics development. And approaching these considerations is not the same sort of linear process. It's kind of more circular, but it's becoming useful. Which button? Okay, so analytic validity basically refers to, does this new test we're developing actually measure what it intends to? What's its level of limit of detection? Is it reproducible? And will it work on the kinds of samples that are generally available? Blood, tumor tissue, fixed in formalin perhaps. A new test that actually can be performed using the standard equipment that already exists is more likely to be readily available at a reasonable cost than something that requires something much more special. Generally, addressing these concerns is the job of the laboratory. It can be very time-consuming, and it's often quite technical. Oh dear, how do I go back? All right, well, clinical validity, clinical utility. These are concepts that address whether or not that information is going to be interpretable and useful in planning treatment. If you measure this thing, does it tell you something about how the tumor is likely to behave or how that drug is likely to work? And for these kinds of studies, we need clinical studies in which patients and physicians participate. And how do we do this? For the most part, we make use of the research infrastructure that we already have, that we've built for drug testing. At least in NCI's Cancer Therapy Evaluation Program, almost all the trials that we support through these networks have built into them a biomarker objective. Typically, the primary objective of these trials is to study the drug, to look at the way that tumors respond to it, how effectively they work. But the biomarker objectives help us develop these diagnostic tools along the way. And the biomarker questions do shift according to where in this drug development pipeline we're at. In the early phase trials, we talk about PD and PK, pharmacodynamic and pharmacokinetic studies, whereas in the later phase trials, we may be interested in whether a biomarker is predictive or prognostic, and I'll explain the difference between those two terms in a moment. Okay. Right. So typically in early phase trials, the primary question is, does the drug work? Does it work at all? So in phase 1, we're asking questions about dose. We're trying to find out if patients can receive the drug without experiencing unacceptable toxicity. And we're asking questions that fall into pharmacokinetics. Typically, these are questions about measuring in the blood plasma how quickly the drug enters the bloodstream after a patient receives a dose, how quickly does the body then break it down and eliminate it. And the pharmacodynamic questions have to do with how is the drug working. So if the drug, say, is some kind of inhibitor of an enzyme, well, first of all, is it getting to the tumor cells? And if it does, is that enzyme actually being inhibited? So these are the kind of questions that are pertinent here. Many of these questions actually require making multiple measurements on what's happening to the same person at various times after they receive the dose. And this can be quite arduous, asking for multiple blood samples or repeat tumor biopsies. And the clinical objectives as well are often focused on the shorter term endpoints. So does the tumor shrink, or does progression happen sooner or later? And the urgency there is that whether the answer about does the drug work is yes or no, we want to get to that answer as quickly as possible. All right. When we get into the later phase trials, where we know that from a phase 2 trial the drug does work, at least in some clinical settings, we're shifting to different kinds of trial designs. Frequently now we're using randomized trials, and survival in one form or another may become the clinical endpoint. And the biomarker question also shifts. We know that this drug works for some people, but really, which are the people who are going to be most likely to benefit? And so we start now talking about, is it, Is our diagnostic test telling us something about prognosis or prediction? In plain English, those two words mean very much the same thing, but when we use it as part of our jargon in clinical trials, we're trying to distinguish two different things. A prognostic marker is something that is sort of intrinsic to the tumor and tells us whether or not it's likely to be more aggressive or more indolent. Things like stage, things like lymph node status, perhaps the number of circulating tumor cells that we find in someone's blood. Not so much related to a specific drug's mode of action, whereas my colleagues who are focused on drug development are much more interested in the predictive biomarker, which is really tied to whether that drug is going to work for this patient. So we measure HER2 amplification to see if an anti-HER2 agent is likely to work. And one of the strategic questions for our partners in the pharmaceutical industry in the later phase trials is whether or not they're actually going to have to budget the time and the resources required to develop a companion diagnostic, a companion to their new drug, a specific test kit that is going to have to go through its own parallel approval process at the FDA so that it's available when the new drug is approved. So I'll go back to the diagram here and just point out that that space in the early phase trials, phase 1 to phase 2, is a very busy one here. I focus on it because this is the phase of trial where people with metastatic cancer are often invited to participate. And it's very complex for the trialists because any and all of these biomarker questions may still be on the table. These are some of the thorny questions that we have to deal with when we're designing trials in the early phases. There's always more scientific information that we'd like to have about this drug. If it doesn't work, why not? Even if it does, do we understand what it's doing? And it's often very hard to focus on the most important scientific question that you can address in a given trial. The question of whether or not we should use some sort of new biomarker to restrict eligibility for a trial to the patients that we think are the most likely to respond. There are pros and cons here that make this question just absolutely agonizing for the people designing the trials. And then there's the question of how much analytic validity do we need? When should we start investing in that kind of research? So usually when I'm at the table, the question that I'm there to ask is if there's a new biomarker being tested here, that's one of the things we're studying. Is there enough analytic validity to justify using it in the trial in this way? And that's never a cut and dried kind of question. Depending on what we're doing, we could be talking about a research test that's being run in a research lab on a few samples that we have from a trial, or we could be talking about setting the stage for a manufacturer's submission to the FDA to get approval for a new test kit that they're going to want to market all across the country. And one of the real questions that we have is that it takes time to get to that analytic validity. You have to start early, and so sometimes we're having to do that before we even know that this particular drug is going to make it out of phase 1. Okay. So here we come back around to these 3 considerations. We want it to be valid analytically so that the information is reliable, and we want clinical validity. It's interpretable, it means something to the patient and the physician in a way that is useful, understanding that that definition of useful really depends on the context. Just a couple of comments I wanted to make for people who may be considering participating in clinical trials. First of all, understand that lab tests are not all the same. Certainly in a clinical trial, you get 1 million lab tests, most of which are the routine kind of tests that you need to monitor your health. But if there's an investigational test whose usefulness is not yet established, then the clinical protocol and the informed consent should make it clear which is which. And if it really is investigational, it may be the case that the lab is able to provide you your actual result. But in other cases, if they're not, if this is really a research level test and this lab isn't certified to operate as a clinical laboratory, they may not be able legally to give you back your individual test result. But you should still be able to find out at the end of the study what the overall conclusion was from your test as well as the others. And then finally, my last comment is that the value of the contribution of people who are willing to provide these samples, particularly multiple samples in the course of a trial, there's just no way to thank people enough for that, because without that, none of the rest of the stuff that I've been talking about would even be possible. So that's where my comments end, and thank you, everyone. And who's next? I think this is the right button to move forward. And you can use this, which is not the same as what's up there.
Yeah, Dan already told me. Thanks. No, I got it. So first of all, I would like to thank Dan and Jill for inviting me here. So I'm really delighted to be here. And my name is Stefan Werner. I'm from UKE in Hamburg, Germany. And our main research focus is the implementation and the development of liquid biopsies. And this, of course, will be my topic right now. So here, the guy on the left-hand side, this is Klaus. So he's an old friend of Dan, but he's also my chief and mentor. And actually, last month he was selected by the Zeit magazine, which is somehow comparable to the Time magazine that you have here, as the Mensch des Monats. That means he's a human of the month in May 2019. And actually, he's a pioneer of the field of liquid biopsy, and together with Catherine Alex-Panabier from Montpellier in France, he invited the term of liquid biopsy. And this was almost 10 years ago in this small review article here. And what we mean with liquid biopsy is the analysis of tumor cells or tumor cell products like DNA or RNA in the blood circulation, but also in other body fluids. And the rationale for this is, normal tissue biopsies, they are invasive and not without a risk for patients. And of course, in some locations, it's quite difficult to obtain a biopsy, like in the lung, the brain, or in the liver. And also, single biopsy might miss relevant tumor areas. So maybe you simply get a biopsy from the wrong position of a tumor. And this might also be better with liquid biopsy because they can represent the whole tumor mass better. And maybe the most important point is that we can do sequential liquid biopsies. So we can take them, since this is a minimal invasive method, several times without any risk for the patients. And of course, the vision behind it that blood-based biomarkers might reveal a more comprehensive real-time information. In general, you can see it on this slide. We are looking for tumor cells, here, the white cells, or for DNA that is floating in the bloodstream. And normally, that should come from the primary tumor or from metastatic sites. And yeah, we get a tube or a blood tube, and yeah, the reason or the more or less well-established technologies or purposes are here for diagnosis, so we can genotype specific mutations, we can follow the response of a patient, so whether some markers are getting up in course of treatment. We can also monitor tumor evolution, so whether we can detect some changes in the molecular landscape of the tumors. And also one important issue, detect minimal residual disease, so whether even if you are operated or treated, whether you still have tumor cells in your body. And finally, this is really an important emerging topic here, which was also presented at ASCO last week, is the early detection of cancer using, especially the sequencing technologies of circulating DNA. Okay, the next slide is quite complex, but it shows you an overview about the technologies that we apply here, because it's a really challenge to find the tumor-specific products in the blood, but because normally you have other things floating in your blood, but not tumor cell or tumor cell products. But we start always here with a centrifugation, to separate the sample where we can find the CTCs or the ctDNA, and for the ctDNA, we have of course to enrich it, and then we can look here for tumor-specific alterations like mutations, translocations or amplifications of specific genes, or we can do this actually unbiased and sequence the complete circulating DNA. So our research focus is more on the CTCs. So this is here the lower part. It always starts with the enrichment of tumor cells. So normally we are using antigens that are presented on the surface of the tumor cells, and these are then captured by specific antibodies. Then we enrich those cells. In the second step, we apply tumor-specific markers or antibodies that can recognize tumor-specific markers. And also for research purposes, we have here the last block where we can also do functional analysis of those cells, try to establish cell lines, or to really do more research-related questions. What is the biology of those cells? So the main application at our institute is here with the cell search system, which was already approved in 2006 by the FDA. And the main principle is that we have, that we have an antibody that is coupled to magnetic beads, and this antibody is recognizing epithelial-specific antigen on top of the cells. And then, the cells were collected or enriched by a magnetic field, and afterwards we apply here a specific antibody that is recognizing tumor-specific markers, so that we can really be sure that this is a tumor cell and not a blood cell. Finally, we end up with this gallery here of cells, so we also have a scientist looking at those cells, and to identify a tumor cell, we need here a cytoplasmic signal, also a nuclear signal, but it should not be positive here for a marker for blood cells. Here you can see a blood cell, and finally you can distinguish here in this gallery between tumor cells and here a simple leukocyte or a blood cell. Okay, the utility or the clinical validity of this test has been proven, especially in early stage cancer patients, so where we have a TNM stage of M0, and our institute has been contributing to a lot of studies here. So we have used it mainly here for breast cancer, but also for other epithelial cancers, and in each of those studies we can show that the CTCs count at initial cancer diagnosis are associated with an unfavorable prognosis. And we have this heard before. So in conclusion, CTC detection improve risk assessment and tumor staging as well as a prognostic marker. Here as an example, this is a meta-analysis that was published last year. So here we have put together 20 or 21 different studies, and we ended up with more than 1,500 breast cancer patients, where we take blood samples and looked for CTCs before start of treatment, and you can clearly see here that we have a poor outcome for those patients who were CTC positive. So this was also correlated to the number of cells we found. So if you could only detect one cell, then it's actually no significant difference here. But when you get higher counts, then this is always a bad prognostic factor. In total, you can also see that in those patients, we find CTCs in about 25% of all tested patients. And the importance of this technology or this idea actually is recognized since 2010 in the cancer staging manual. So where we now have an own staging classification with lies between here M0 and M1. Yeah, actually it's called M0I+, and this indicates that we can find individual cells either in the bloodstream or in the bone marrow of the tested patients. So what is the relevance for detection of minimal residual disease or for post-surgical surveillance? There I would like to show you this slide here. So here's plotted the tumor load to the progression of the disease or the time course of the disease. And actually we have two thresholds here. One is for the minimal residual disease, and the other one is for the clinical diagnosis of the disease. And normally we can only detect, or with conventional methods, we can only detect the tumor when it reaches a certain mass. And of course, then the treatment starts, and there's a good chance for cure at this point. But in some cases, we get minimal residual disease. And actually, our idea would be that we can use our technology also to identify those patients with MRD in order to taken up treatment again and hopefully reach here a cure in a second-line treatment. And we have also a study here published last year with contribution of our institute, where patients were analyzed 2 years after chemotherapy. And as you can see here, also those patients who have CTCs in the bloodstream, they have a much worse prognosis than those without CTCs. So this is, I guess, a very important application, because maybe we can use these technologies in the follow-up period to identify those patients whose therapy is not working. Okay, and finally, I would like to show you some examples or some possible applications for metastatic patients and the early assessment of therapy responses. So, of course, in the meantime, or in the last decade, we have, the research community has developed a lot of targeted therapies that aim to inhibit specific cancer drivers here, like KRAS or EGFR. So, of course, for breast cancer, we have also a lot of substances that tackle the estrogen receptor, and the same is true here for prostate cancer, where we have anti-hormone therapy like in breast cancer. And for this, there was also a very interesting study published 2 years ago where not only the CTC as such was used, but also what is expressed in those CTCs. And in this case, they used a splice variant of the androgen receptor, which is, of course, the target here for the targeted therapy. And here in this plot, you can see that I have to show you this. Each column here represents the response of an individual patient, and this is measured by the PSA response, so whether the PSA serum level is going up or down, and when it's going down, then you, of course, have a positive response to the treatment. And with patients where we can find here no CTCs, they almost responded quite well. When patients had here CTCs in the bloodstream, that was already not so good anymore. And here on the right side, you see those patients that had CTCs and on top of that also express this specific splice variant of the androgen receptor. This splice variant confers resistance to therapy, and consequently also those patients do not respond that well. Okay, taken together, CTCs and circulating DNA provide complementary information as liquid biopsies. It's also complementary information compared to the normal tissue biopsy. So I guess we can never replace a normal biopsy, but of course we can deliver additional information with these technologies. But by now we only have a few really good validated assays. So, So we still need to validate a lot of assays also by independent research group. We have a lot of papers that were coming from single institutions, but of course the tests have to work at different research sites. And maybe the most important topic is how to integrate now this knowledge or these technologies into interventional clinical trials. Because by now we can only improve the diagnostic of the patients, but of course, finally, we would like to help the patients and maybe to make treatment decisions based on results from the liquid biopsies. And to do this, we have actually or are planning to have a European liquid biopsy society, which is initiated by Klaus, and here we have the clear aim to integrate liquid biopsy into a clinical practice. And finally, I would like to thank here our research team in Hamburg, and we'll be happy to take your question afterwards.
Okay. All right, thanks for having me. My name is Chad Pico. Thank you for Dan and Jill for inviting me. It's nice to come to a talk that took me 10 minutes to get to. So my lab is across the way in the medical center. It's always fun for me to be able to give a talk in front of cancer patients and survivors, because for me that's where it all started. So I am a cancer survivor to begin with. So this might be a little bit unique that my perspective comes from a prior cancer patient, cancer survivor, cancer advocate. I am now a cancer doctor. I ran to dinner from clinic last night, and I'm a cancer researcher. I run a research lab as well, studying cancer, and my focus really is on metastases. My motto in my lab is, if it does not spread, I don't care. So you'd be amazed how many cancer models there are. How many cancer models there are that don't spread. It's actually remarkable how few cancer models people study just simply do not spread. I could talk an hour about that easily. So my talk is about imaging. But this is actually kind of an easy talk to give for me because everything I do in my practice is based on imaging in large part, as well as molecular diagnostics and biomarkers. We do a lot of liquid biopsy now. But there are other reasons why this is kind of an easy talk to give, because I'm just going to show you a bunch of pictures. There are very few words in this talk because I really think what's in the pictures, and obviously it's sort of implicit, this is imaging. There's a lot more information in these pictures than anything I could put in bullet points. I also hate bullet points in slides, so I don't put them usually. This is actually a graphic that I drew when I came to UNC. It's still not been published yet, but I think it illustrates an important point. And it's really, there's really kind of two important routes that we think how cancer spreads. Most metastasis reviews focus on the bottom part, which is, let's see, I'll get this laser works. Not good. So basically the bottom part is how most metastasis reviews describe how cancer spreads through a bloodstream. The top part is actually more meant to be illustrative for the purpose of the imaging talk. But cancer spreading to lymph nodes is something we've known about for every single cancer for well over a century. It's very poorly studied. Until last year, there were 2 papers in Science that came out in the same issue showing that cancer can leave lymph nodes. But until that came out, we actually never had evidence of that. Most people just sort of assumed it. But what's more important is that we know that cancer can get to lymph nodes. This pointer is not going to help. The cancer can get to lymph nodes, and so on scans, we look for lymph nodes to see if they're enlarged. And certainly they can go to any other location. And certainly the natural behavior of cancer depends on the cancer, but some cancers really like going to the bones. Some cancers really like going to the brain. There are also some really weird idiosyncratic cancers. I'll give you one random example. Ocular melanoma, literally melanoma in the back of the eye. Just curious if anyone has a guess where that almost always goes to. I wouldn't expect anyone to know. Melanoma that starts in the back of the eye, 90% of the time goes to the liver. Pretty bizarre. So certain cancers have a predilection for certain locations of spread. And I think probably the person that hit on that the best first was Stephen Paget. So the Seed and Soil, it's actually hard to find that paper. I actually have it. It's from, I think, 1880. And he did an autopsy on something like 500 or 600 people with cancer, trying to understand what's unique about certain cancers and where they spread. And that information, a priori, helps us. So as a physician, when I look at scans on patients, I usually have an idea of, all right, do they have lymph nodes that are enlarged? Do they have brain mets, bone mets, and so forth? All right. So I told you I'm not going to show a lot of bullets, and so for the sake of time, I'd rather just show you the pictures. Does everyone get these slides? Good. All right, so you can read these later. These are more definitions of how these scans work. A CAT scan is actually a fairly simple technology. All it is a bunch of X-rays going through the body, circling around the body. And essentially, using an algorithm, the computer essentially generates an image. But all it is a bunch of X-rays going through the body as you go through it. So let me show you a CAT scan. I think some of the pictures I chose are sort of illustrative of the challenge I feel like the patient has and the physician have in that interaction describing what we're seeing on the scans. And so I felt like I would spend a little bit more time on what are these scans actually showing. So what I did mention on the last slide, if you read it, essentially is that on a CAT scan, the wider something is, the more dense it is. The darker something is, the less dense it is. And so when you look at a CAT scan, what should jump out at you are 2 things, sort of gradations of white and dark. So if you look at lungs, they should be pretty black. And if you think about it, for pretty obvious reasons, lungs are mostly just air. There's not much density in the lungs. There's actually alveoli and air inside your lungs. Now there are some dense things in the lungs that should be there. Let me see if this will work. No. So blood vessels, there's a lot of blood vessels here in the lungs. And so the white areas going through the lungs that are normal are blood vessels. If you think about blood, it's dense, and so that's actually pretty normal. But clearly there's, on your left, you see a big tumor. I hope y'all know, can y'all see my pointer? Yeah. All right, so y'all can't see, I can barely see that. So here is a big tumor. So it's dense, it's actually pretty white. Just to give you an example, this right here is bone, that's the spine. And bone, as you imagine, is really dense, so it's white. So this is actually relevant for what I'm showing because if you look at this tumor, and I just want to kind of have you just try to picture this sort of being you. If you had a tumor in you, and I'm telling you, all right, well, here's the tumor, and then we put you on treatment, and let's say whatever the treatment is, this is the next scan we get on the right. here's, I think, a really big challenge, and that's why I chose this image. That tumor actually didn't shrink much. If I gave you the dimensions across this tumor here and across here, it's possible actually in some areas, if I measured it in some areas, it would actually even be bigger. But what's pretty evident, if you look at the center of the tumor, it looks very different, right? So now it's black. And like I said, because of density, we know that's just air. That means that tumor responded dramatically on the inside. It's essentially gone. All the tumor in the center is gone. And so if I saw that in my clinic, I'd be actually pretty thrilled. This patient had a phenomenal response. But the radiologist, most radiologists, at least the way they read scans, are boring people. So sorry if there's any in here, but all the reports are usually very mundane. They're very hedgy. They're avoiding lawsuits. They almost never say anything exciting. There's no fun superlatives. But this should be like, wow, that's awesome. The way a radiologist would read this, and so if you got the scan report, it would be like, well, minimal change in dimensions, right? Because there's not much change in the dimensions, but there's a dramatic response. There's a dramatic change of what's going on in that tumor. Conveying that to patients I actually find hard, because I have some patients that will read the report immediately when it comes out, even after I just spent an hour in my clinic with them. So that's actually why I take a little bit of time to tell you all about it, that sometimes the radiology report doesn't tell you everything, and sometimes actually having a little bit more savvy with what's going on in these images is helpful. This is actually really exciting. It's possible there's no tumor left. It's possible all that's left is scar tissue. Let me show you something that's not just scar tissue. So this is a metastasis meeting. I felt like this is relevant to show. This is actually really hard, I find, to explain to a patient. Now again, I've been a patient, I do research, but when I'm in my clinic, I'm trying to think, well, how do I convey this to the person across from me, what's really going on? And I hope they don't read the radiology report in this. What the radiology report here would say is innumerable. You can't number them. There are probably thousands of metastases here. It's kind of scary. And I can tell you, I'm not going to tell my patient, there are thousands of metastases in your lungs. It's scary, right? I would probably say something more along the lines of there's quite a few tumors there. We really need to do something about this. But that's scary. I mean, can anyone here feel like they can really count that? And how would you relay that information to somebody? That's alarming. This is looks like snow, right? Those are all metastases, and that's all what we can see. Actually, a little bit to Stefan's point, he showed a slide that actually illustrates something that's really, I think, quite helpful. Scans have gotten better and better, and that's actually a little bit of a concern. Scans have gotten so good, we actually see more than we used to be able to see 5, 10 years ago. And so we see more and more things now that we're not really sure are those cancer or not. But let me tell you something that might alarm you, and it should probably a little bit. A lot of these little tumors here are about 5 millimeters in dimension. We now have the ability to see metastases down to about 2 to 3 millimeters in dimension. That sounds pretty sophisticated, but there's actually something you should know about that. A tumor that's only about 0.5 centimeter in size represents about 10 million cells, cancer cells. A tumor that's 1 centimeter in size represents closer to 100 million to up to 1 billion cancer cells, just in one little tumor. So what I'm telling you is most of these things have up to 10 to 100 million cancer cells in each one of them. So how far down do you want to be able to see? Do I want to be able to see down to the individual cancer cell? I'm not sure, because eventually if we can do that, imagine how hard that'll be to convey. Now we see 10 to 100 trillion cells in your body. That's a little alarming for a patient to hear, but eventually reports will be generated that actually that sophisticated we can tell you exactly how much metastasis you have. Is that relevant? I don't know. Maybe that'll be good for the Q&A. All right, so PET scans. So you want a PET scan every 3 months. And so here's what you get with a PET scan. That's actually really interesting, by the way. I never had a patient want PET scans every 3 months, so I would like to actually talk about that if you're interested. So what do PETs do? And then I'll talk a little about a PET CT scan. So a PET scan is potentially even equally or even more challenging sometimes to interpret, but essentially what a PET scan is looking at least our current modern day PET scans, are just looking at how much sugar activity, consumption, is taking place inside of a tumor. So if you think about cancer, the more metabolically active and fast-growing it is, the more it desires sugar, and it takes it up. And so the PET scan sort of exploits the ability to be able to see what tissues take up lots of sugar. Well, I'll tell you a couple other areas that hopefully are taking up a lot of sugar in your body that are not cancer, and that would be your brain. So if it's functioning a lot right now, it's taking up a lot of sugar. And so when you do a PET scan, the brain is always glowing, and I'll show you that. If you think about other areas, the liver consumes a good bit of sugar. The heart consumes a lot of energy, and so the heart always lights up. And then actually to be able to see this, it's actually kind of a fake sugar molecule. You have to give the contrast, so it goes in the IV. Well, guess what? You also pee that out. So the bladder always lights up because the kidneys and the bladder always light up because they're basically dumping all of this contrast. So let me show you an example of that. All right, so everything's lighting up here, right? Lots of things lighting up. But let me show you which things are relevant in terms of a cancer patient and metastases. So the brain lights up, and that's good. If that did not light up, that means there's no brain activity. So that, fortunately, you want to see light up. And then here's the bladder, so that lights up. So these things we expect. Here's the heart, and that lights up. So essentially, if a lot of other things are lighting up, aside from what I just pointed out, there's more metabolic activity in the body than you want. And that should be pretty evident looking at this. So look at this. This is a bone metastasis in the spine, here and here. See how bright those are? Here, this is lighting up way more than the heart would, and that's concerning. That's probably the primary tumor where it all started. And that's all I'm going to point out because that's all that's really necessary. These are areas that light up. But here's where it's challenging. So let me show you an example where it's challenging. So this is what we call a PET CT. You're literally overlaying a CAT scan and a PET scan together. It's actually a really helpful scan because it gives you information on structure of a tumor as well as how active it is metabolically. And so let me show a little bit of this. All right, so this is an area that's not normal. That should not be in the lung. And here it is on the PET scan. You see it lighting up. I actually don't know because I just got this from Google Images, whether that's cancer. But let me tell you, I actually couldn't tell you from that image if that's cancer. Why? Because I see something that's abnormal in the lung and is glowing. All that tells you is a couple things. There's a structure there that's not normally there, and it's metabolically active. Well, let me tell you what else can look like that in the lung that's metabolically active and a structure shouldn't be there. Scar tissue, pneumonia, some other kind of inflammation for some other reason. So things that are metabolically active that end up showing things on scans that are not supposed to be there still don't guarantee you that's cancer. Now, it might be a little concerning. The only way you're really going to know in this case is to get a biopsy. So, and this is maybe helpful for the Q&A to kind of elaborate on this, but I think that's some of the challenges we have in the clinic. The scan technology is good, but it's probably not nearly as sophisticated as it's going to be in maybe 5 or 10 years. And the question is, as it gets better, what information do you really want to know? As I'm saying, eventually we might be able to tell you way more metastases 5 years from now than we see now. But think about that. If I can see, let's just say, 5 metastases in your lungs now, fast forward 5 years from now, if I could see 100 more metastases 5 years from now, that doesn't change where you were right now clinically. It's still going to give you treatment for it. It just tells you how many more things I can see now in your body. And I do wonder, actually, sort of being on both sides of the fence, how much that information is going to help us. I don't know how much it's going to change how we take care of you or your loved ones. So those are things I think that as the technology gets better, I really have to wonder about how we actually convey that information and how we actually use it. All right, radiomics. This is kind of a field I didn't believe in when I first heard about that, and that was actually only like a year ago. So I was at World Lung Cancer Conference in Toronto, and I saw a couple posters on radiomics. I'd never heard about it before. And actually, I really think this is going to be something quite interesting. And like many fields, for example, molecular biology, people want to know about their DNA sequencing. That's actually become a bit of a problem for pathologists. Pathologists are used to kind of dictating everything and how treatment decisions are made. And now I can get more sequencing information and value on a tumor than a pathologist could ever give me. That's a problem for them. This, I think, will be a problem for radiologists. Radiologists want to be able to tell you what they see on the scan and bill heavily for it and stay in their darkroom. What's interesting is this actually is a field that's going to change. This actually tells you that you could take a scan. So in this case, here's a scan, here's a CAT scan, and instead you use a lot of machine learning, and essentially you extract a lot of sort of digital information. So here's all the digital information being extracted from a single scan, and it's actually kind of nuts how much information a computer and softwares can actually extract from a single scan way beyond the human eye can extract. And what's surprising is a lot of the information coming from the radiomics is actually telling you biological behavior of a tumor. To give you an example, and again, I was very skeptical when I first saw this, a CAT scan like this can now be used to predict whether your tumor responds to immune therapy. Never would have believed that. But the data coming out is actually quite compelling and interesting. I did put a citation here, so if people have an interest in this, this guy here is pretty impressive and is putting out a lot of interesting papers along this realm. So what does this mean? It essentially means you can go in, get a scan. Eventually, the scan will be imputed into a computer. Lots of information will be extracted from the scan, and then it'll say, here's all kinds of biological information about that tumor, and basically totally steps over the radiologist. So that's a problem they're going to encounter soon. All right, so again, kind of drilling down to how far into the tumor, how much do we really want to know as far as metastases? How well do you really want to be able to see metastases? So this is kind of this is a paper that came out this year. It is just in mice, but I do believe this is going to be sort of the future we're going to see in terms of how good the imaging is going to get. So I showed you PET scans, essentially a tumor lighting up based on how much metabolic activity it has. On the left, something called bioluminescence. It's sort of a similar precedent. It's kind of crude, so you can imagine this being kind of a PET scan in a patient, your ability to see tumors and metabolic activity. What you see on the right, now keep in mind this is just in mice. It is the kind of technology we have, but it's using something called light sheet microscopy. The technology itself is not important. The point is, I think, more important. This technology is so unbelievably phenomenal, it allows us to now see down to the individual cell of metastases. I think on the next slide I can show that better. So what they're seeing here is thousands and thousands of lung metastases, stuff that you would never see in a CAT scan. In fact, if this were in a CAT scan, you probably wouldn't see a single one of them. But there are thousands of lung metastases in this mouse that light sheet microscopy could see. On the left, you're seeing a 3D rendered image of this, and on the right is just sort of a single slice. What does that mean? Eventually, the better signal we have to be able to detect small amounts of cancer cells, the more likely this is going to become a reality, that we'll have non-invasive approaches where you can go into a scanner, and we'll be able to see down to the individual cancer cell. That's actually a little scary to me. I don't know how I would explain to a patient, we now see 2,000 tumors in your lungs, when if I just did a CAT scan, I might see 3. And that's sort of what I'm getting at. I don't know how relevant that's going to be with how we treat patients, but it might become super relevant, and it may become super sophisticated. Maybe our way of relaying information will have to grow in sophistication, and certainly what I have seen in my clinic is patients know a lot. So I got to say, I guess I became a de facto cancer advocate 19 years ago when I had cancer. Patient advocates now are very different. I feel than when I became I just cook meals for cancer patients. You guys are a force, and it's actually very cool to be able to see sort of all the sides of it. And it kind of makes me stand up a little straighter and be like, wow, they're really on it. They know a lot of stuff and they have high expectations for the future of cancer research. So I think that's neat. And so I think as patients become more informed of their own care and the field itself through forums like this, we're going to have to understand how do we better understand cancer metastases in terms of imaging, and how do we explain that to each other, and how is the response going. So one last point I'll make is, what if 2,000 out of the 2,500 tumors are responding, and the other 500 are growing a little bit? How do you explain that? How do you resolve that? What does that mean? So these are kind of questions that I would actually put out there to kind of be interested in knowing how well do you want to know about all the metastases that we can see based on imaging. I think that was my last slide. Yep. Thank you.